Thank you so much for joining us. I am delighted to be here this afternoon to discuss stem cell-based therapies for age-related macular degeneration, a disease that, as I'm sure you all know, affects 11 million people in the United States. Yesterday, we were absolutely thrilled to announce that at NYSCF, we're developing a therapy for AMD in collaboration with scientists and clinicians at Columbia New York Presbyterian Hospital and the National Eye Institute. And the goal is to improve or restore or stabilize vision for patients with macular degeneration. And at NYSCF, we recently completed construction of a specialized facility, which is called a GMP suite, to manufacture the cells in a safe and robust way so they can be used in this therapy. Joining me today to discuss this work is our esteemed collaborator, Dr. Stanley Chang, professor of ophthalmology at Columbia University Medical Center, where he led the Department of Ophthalmology as chairman for almost 20 years. Dr. Chang is a specialist in vitreoretinal disorders and surgery, and he pioneered many of the surgical techniques currently used in this field. He continues to improve clinical outcomes for people through his visionary research and innovation. Also joining me today is Dr. Kapil Bharti, a senior investigator at the National Eye Institute of the NIH, where he leads the unit on ocular stem cells and translational research. We will hear in a bit from Dr. Bharti about what is the first ever stem cell trial for AMD in the United States that uses stem cells created from a patient's own blood to create the cells for transplantation back into a patient. This trial is for macular degeneration and is a huge milestone in not only the field of ophthalmology, but stem cell biology writ large. We at NYSCF are collaborating with Dr. Bharti, and we're utilizing his technology to help advance our therapy to the clinic as fast as possible in collaboration with Dr. Chang and his colleagues at Columbia. We are so grateful to be working with these amazing scientists and clinicians, and I am delighted that they're both joining me today. And I just want to take a moment to acknowledge that our tremendous progress in advancing this therapy, where it is going to be a reality for patients, has only been possible because of visionary support from our philanthropic partners. So to ensure that we get across the finish line as quickly as possible, we still do need additional funding. So if bringing this therapy to patients with AMD is personally important to you or a loved one, we'd love to talk with you about the vital impact that you can make. So please let us know through the chat or reach out to us afterwards. And before we begin, I want to also thank the Stavros Niarchos Foundation for their ongoing support of our education and outreach program like today's event. And we also have a matching grant challenge from SNF to help ensure we can continue this work. So if you have enjoyed the discussions, please let us know that SNF will be matching your donation one-to-one, doubling the critical impact on the programs for the public and students as well. So to get us started, Dr. Chang is going to give us an overview of what age-related macular degeneration is, both in the wet and dry forms, and the currently available therapies and the limitations on those therapies for patients. So, Dr. Chang.
Thanks very much, Susan, and thanks very much for including me in today's conference as well as in this program, which I think is going to revolutionize the way we can treat macular degeneration. With Kapil's expertise in and experience in cell transplantation and bringing this into the clinical arena, I'm pretty excited about the whole prospect of using stem cells for the treatment of dry macular degeneration. I'm going to cover the basics of macular degeneration. There's a diverse group of audience today, so I thought I'd cover some of the points, both from the epidemiologic side as well as scientific side of what's going on now. As you know, age-related macular degeneration is a leading cause of irreversible blindness over the age of 60. It does affect women more than men because women tend to live longer than men. And that by 2050, the affected number of Americans affected will be about 5 million. Some have estimated higher in comparison, and it's more than double of what it is currently. In addition, the current treatment with intravitreal injections of medications have become the most frequently performed eye procedure globally, not only in the United States, but globally. And this procedure is done in about 10 million Americans a year. Here's a patient typically that comes in with early changes of macular degeneration. Often they don't have any visual changes here. Her vision's pretty good at 20/25. And you see these yellow spots in this picture of the retina, this roundish on the left is the optic nerve, and in the macula, which is in the center of the picture, you see that they're relatively clear, but it's surrounded by these yellow dots, which are deposits composed of the chemical action of phototransduction, and they consist of lipids, lipoproteins, amyloid, and other inflammatory proteins in the retina, and they're deposited on the undersurface of the retina itself, on top of the under the RPE. And when large numbers of these yellow spots, as in this patient, appear, there's a greater risk of macular degeneration. And what are the other factors for macular degeneration? Age is certainly a factor. You see that as the decades increase, there's almost an exponential rise in the occurrence of age-related macular degeneration. And it tends to occur in Caucasians more than non-Caucasians. Although Hispanics can be also is increasing in Latin. And blue irises tend to have more likely to develop macular degeneration. The other factors are well known, family history of macular degeneration, and there are two genes, the complement factor H and the ARMS2 gene, which are the most prevalent genes causing about 70% of cases of macular degeneration. These two genes, by the way, are tested by 23andMe. When you look at risk factors for different diseases, 23andMe does test the incidence of the presence of those genes for you. However, they don't tell you if there are any protective genes, so it doesn't explain everything about whether you would get macular degeneration. Smoking is a risk factor that increases the risk by 4 to 10 times higher. Obesity and high glycemic foods, and a history of systemic hypertension or blood pressure elevation. We're very fortunate now to have very good diagnostic techniques to detect the retina with high precision. And we have a photography on the top with these color images of the retina, what we call a 50-degree field, but also wider views of the eye. The macula only involves a small portion of the retina, but you can see how much more retina there is beyond the macula, and that gives you the peripheral vision and the night vision. The macula gives you all the close and reading vision. And then we have this test called OCT, which has a resolution of up to 5 microns. You see the different layers of cells within the retina. Here's a normal eye with normally a little dip in the macular area. And just this one central area gives you all the reading vision. Oops, sorry. And it gives you all the reading vision. And many times we can see changes in this scan before the patient notices any change in visual acuity. So we're fortunate that we can detect very early disease and very early changes. Macular degeneration is broken up into two types: dry, so-called dry, where there's a degenerative process causing atrophy of the retinal pigment epithelial cells, these orange layer of cells in the background, because the retina is transparent, and the photoreceptor cells. The visual acuity loss tends to be relatively slow, and there's currently no good treatment. Here you see in this picture that the central area, there's no orange pigment. That means most of the melanin and the RP cells have disappeared. On the right, you see the wet form of wet macular degeneration, where there's growth of leakage from blood vessels under the retina. And this retina causes fluid to leak and sometimes bleeding under the retina and causes loss of transparency of the retina, as you see here in this hazy view. This whitish tissue is the scar tissue and blood vessels that grew under it. Both can cause loss of reading vision, central vision, and you can see that actually the dry macular degeneration in this case was worse than the left, than the eye with the left. But the rapid, but wet macular degeneration is more likely to occur more rapidly. So we mentioned that there's no good treatment of dry macular degeneration. So ultimately, if this involves both eyes, we have to use magnification and increased contrast, something like an iPad to help or a Kindle to help patients read and increase the size of the font size so they could read. But ultimately, when they lose central vision to 2200 or worse, they have more difficulty managing. About 8 to 20% of patients convert from dry to wet macular degeneration. So we have to watch for that as well. And if it does convert to wet, we can use the treatment for wet macular degeneration to try to stabilize the condition. We also know that long-term treatment of wet macular degeneration over the years, after, let's say, 10 years of treatment with injections for wet macular, they can progress. Patients can progress to loss of vision, lose further vision from the dry component of macular degeneration. Here's a typical patient with wet macular degeneration who had a bleed under the macula. You see the dark blood involving his central area here. And after several injections of anti-VEGF, the blood disappeared and leaving a scar. But his vision improved a little bit and he could had a greater field of vision, but he still couldn't read. And this is how we do the injection. We do it in the office. A needle goes through the side of the eye here and into the so-called vitreous cavity. It's done under anesthesia and topical preparation in the office. Here's the appearance of a typical patient. It's surprising he has maintained excellent vision after 10 years of treatment of macular degeneration. But you see on the right-hand side that the scans that show the blood under the red arrow here, and as the treatment has been given, the blood reabsorbs, leaving only the scar present. Now, there are several different drugs available. They all inhibit vascular endothelial growth factor, which is a protein that causes leakage of blood vessels and growth of blood vessels. And the drugs generally last between 1 and 3 months, and they have to be repeated every 1 to 3 months, usually between 1 and 2 months. Because there are so many people affected by this disease, these drugs have become among the top 3 drug expenditures in the Medicare budget. So this has become a very frequently researched topic by industry because they feel that there's good potential for developing a good business with this drug. And at the same time, they have helped a lot of people because it's really changed them from becoming totally blind to becoming somewhat able to use reading aids if they can limit the progression of the disease. We also have a new technology called OCT angiography to follow the growth of these blood vessels and visualize them. Here on the left, you see the OCT scan. This bump is where the blood vessels grow. But in the past, we used to visualize this by injecting a dye called fluorescein angiography, and we inject it in the vein, and then they take pictures of it. The patient turns orange, and his urine turns orange for a day or two. Sometimes they they don't tolerate the injection too well. But here is a picture of the OCTA showing this blood vessel growth under the OCTA, right under the macula. And this can tell us whether this membrane is getting larger or smaller over time. And this can be done now non-invasively, and usually can be done almost every visit. So the problems with current treatments is that we have to bring the patient back every 4 to 8 weeks for the treatment. And each time we give an injection, there's about a 1 in 1,000 risk of infection. So over a patient's lifetime, the risk of infection is pretty significant. And then after long periods of treatment, patients may lose their visual acuity from the progression of dry macular degeneration. So our goal is to maximize the benefit of treatment while reducing the treatment burden and the risk of injection. So many other drugs have been tested, and I just want to mention a couple of them. Some of the emerging technologies that are about to be introduced in this field. Okay, so one of the technologies that just is finishing its evaluation is called the port delivery system. This is an implantable reservoir that allows a drug to be kept in a small chamber and slowly released over a period of 6 months. It does, however, require a surgical operation. You see the picture here of it in the eye, covered by tissue. Clinical trials recently completed in phase 3 showed that there's non-inferiority to monthly injections. So compared to monthly injections, the patient only needed to get injections every 6 months. However, because it involves an operation and a device that's implanted, there is the potential that this tissue could erode over the surface of the device and cause an infection. And also some of those devices have been known to be dislocated into the eye. When you put a needle into it, all of a sudden it falls back into the eye. It's uncommon, but these are potential complications. A new drug that has been almost finished its phase 3 trial is Furisamab. This is a bispecific antibody. This binds two components. One is a component that binds the vascular endothelial growth factor, like in other drugs, but a second component binds angiopoietin 2. And this binds Tie receptors in the tyrosine kinase pathway that reduces vascular permeability and increases vascular stability. And in recent studies, they've shown that this, in many patients, they could go as long as 4 months, and at the same time showed non-inferiority in macular degeneration compared to the most frequently used drug, Eylea or aflibercept, injected every 2 months. So this drug may offer a longer effectiveness in the treatment of wet macular degeneration. Another approach that people are using to try to treat wet macular degeneration is gene therapy. And this drug developed by REGENXBIO uses an AAV or adenovirus vector to deliver an anti-VEGF fragment, antibody fragment. The gene codes for the anti-VEGF fragment. It's encapsulated incorporated into the vector, which is an adenovirus. Adenoviruses causes pink eye, as you know, and it sometimes causes epidemic keratoconjunctivitis, but this is injected into the eye itself, and there's no infection, but it can cause a little inflammation. And this is incorporated into the retina. Unfortunately, it has to go under the retina, so we have to do an operation where we remove the vitreous, and then we take a small needle and insert it into the retina, under the retina, and inject a bleb of fluid with the virus in it, and this transfects the RPE that releases the FAB fragment of anti-VEGF. And this vector is delivered through the retina. And early studies show only a small series showed 14 of 24 patients did not need any further injections, and the disease activity was stabilized. Another gene therapy company called Adverum has developed a vector that you could introduce into the vitreous in the office. So this is not injected under the retina, but into the vitreous of the eye. And this AAV, or adenovirus variant, can penetrate through the retina as well as into the RPE and release a aflibercept into the subretinal space. And some of the preclinical trials and the early clinical trials are very encouraging. You can see that when this vector is injected into the eye, where the green is, this vector can be expressed. VEGF can be expressed. The anti-VEGF could be expressed throughout the retina and throughout the eye. And you could see also in their early studies, in phase 1 or phase 2 studies, that there's been quite a significant reduction in the number of rescue injections that are needed to save the acuity and stop the leakage in the eye. So this is a promising technology. However, there are still many questions about gene therapy. How long will the gene therapy last? And if the patient develops antibodies to adenovirus, can the treatment be repeated when it becomes necessary? So is it only a one-shot type of treatment, or will it have to be repeated? And if the patient has antibodies, can he take another injection without building an immune response? So let's talk about dry macular degeneration. So far, the only treatment for dry macular degeneration is vitamins. And many of you take the AREDS vitamins who have macular degeneration, take vitamins. It probably does not have a strong effect treatment, but has a moderate effect on the treatment of the development of macular degeneration. And so there are really no current highly effective treatments. And the new treatments that are pharmacologic are aimed at altering the complement pathway, which is one of the genes that cause macular degeneration, the complement factor pathway. And at best, all of these treatments would slow the progression. Here's a picture on the left, and you see the general grayish background of the retinal pigment epithelium, but right in the center, it's white because there are no cells, RPE cells, covering the macula and supplying nourishment to the macula. So one of the companies blocks this complement C3, which is a major component in the complement activation pattern. Both the lectin pathway, the classical pathway, and the alternative pathway are all blocked by C3. And this drug is injected monthly or every other month. And you can see that even with the injections over the course of 12 months, there's only a 20 to 29% chance that the progression of the atrophy and the dry macular degeneration would be slowed. So is it worth it for someone to take a lifetime of monthly injections to slow a process with only a 20 or 29% chance of slowing the process? In most cases, most of my patients wouldn't go for it, except for some patients where the second eye is getting affected. They have no other choice and they want to try to do everything possible. This is one option. Another is the so-called complement factor I. And complement factor I is an inhibitor of the alternative complement system. But the deficiency is only present in about 5% of patients with dry AMD. So doesn't include all patients with AMD. Only a small percentage can get this treatment. And the gene therapy is aimed at upregulating the production of complement factor I. And this is also injected subretinally. Here's a picture. We take the patient to the operating room. We inject with a small needle under the retina, and a little bubble of fluid forms under the retina, filled with this vector. Another approach that they're evaluating is the subchoroidal method. Here's a little tube injected under the wall of the eye, under the retina, and a needle is extended, and that needle contains fluid that, contains the vector. And once the needle penetrates the choroid and into the subretinal space, the surgeon injects the fluid, and a little bleb of fluid with the vector is injected into the eye. So these would only stop the progression of dry macular degeneration. So for stem cell treatments, I think this offers the possibility of actually improving the vision. There have been previous reports and experiments where suspensions of embryonic stem cells have been injected subretinally. Patients were treated with steroids to suppress the immune system so they wouldn't reject these cells. And older patients with macular degeneration don't tolerate steroids very well. Pete Coffey, who's a member of the board with Lyndon de Cruz, also reported 2 patients in London where they did, who had sudden loss from wet AMD. They suddenly removed the scars and the blood and put embryonic stem cell transplants with improved or stabilized vision. And there have been small groups of patients treated in California and Japan with iPS-derived RPE cells. So Susan with the Stem Cell Foundation and Kapil with the technology that he's developed has really developed it to the point that we can have a larger clinical trial throughout the country. And I think this offers the opportunity to really change the way we can do things for these people, because the late stages of macular degeneration, whether it's dry on the left, on the right, or wet on the left, the so-called disciform scar, they really cause people to lose their ability to function, to socialize, to recognize people. They're unable to read, they're unable to drive, they feel isolated. The loss of sensory input may accelerate dementia. There are many of my patients who have end-stage disease who complain of hallucinations, and they wonder why they get hallucinations. This is quite common with the Charles Bonnet syndrome. And despite any low vision rehabilitation service like magnifiers or accessibility software, they're just struggling to maintain some quality of life and ambulation so that they can continue to function on a reasonable level. So I hope that Kapil is going to show you that the possibility of restoring and regenerating the macula in dry macular degeneration cases with his beautiful work.
Thank you so much, Stanley. That was really comprehensive and really appreciate it. And I think everyone understands how urgent the need is, and the great hope that we have with these trials. So Kapil, you've been a pioneer in this field and now are developing one of the first pluripotent stem cell therapies in the world. I think it's the first in the US that I'm aware of that is an autologous pluripotent stem cell trial. The autologous means that you're using blood from the patient so that when you make the cells, it will be the patient's own cells. This is the same type of therapy in development at NYSCF. I'd like to ask you to just talk a bit about the advantages of making a cell therapy from a patient's own blood sample, and also the the state that your trial is in right now, because I know we have many, many questions. And one of these is, how do I sign up for a trial? And your trial is going now. So we're collaborating with you, and we're incredibly grateful to be, making cells for transplantation and to be working with you on this incredibly innovative technology. So let's talk about what's unique about your process, and how this is going to help clear the path forward for our efforts at NYSCF.
First of all, thank you, Susan, for having me here and listening to Stanley about all the problems these patients face. It's really heartbreaking. And thank you, Stanley, for your kind words about our work. So autologous, like you said, it means patients on blood, starting from patients on tissue, be it blood or fibroblasts. In our case, it's blood cells. In your case, also it's blood cells, as opposed to allogeneic, which would be somebody else's cells back into somebody else. So the advantage of an autologous tissue would be that it's immunocompatible. It's patients on tissue back into the patient. And that means we don't have to give them any immunosuppression during or after the surgery. And as Stanley would tell you, these are old patients. These immunosuppressions have very bad side effects onto these patients. So by doing this autologous process, we avoid all of those adverse events or side effects of those immunosuppressive agents. Now, to develop an autologous cell therapy, what is a key point that you need? That means you have to be able to make this tissue from every single person over and over again. So your process has got to be very robust. It's got to be very efficient. You got to be able to make it every time successfully. And you got to be able to QC it, quality control it, and say, yes, it's ready. It is transplantable. I know it will work. And I think that's the key. That was a unique part of our process, that we spend a lot of time making sure that our differentiation protocol, as in from iPS cells to the RP cells, the cells that you see behind me, the cells that are damaged in AMD patients' eyes, that this process works. The protocol is absolutely perfect in terms of making robust, fully differentiated, fully matured, polarized cells that would, once transplanted in the eye, would so-called hit the ground running and start functioning immediately. So that was a key point, right? So, and I think that really will help in this collaboration that we have set up between NEI and NYSCF in Colombia, that once we have transferring, now we have successfully transferred parts of the protocol and we're doing rest of it as we speak. As we fully transfer the protocol to you, you will be able to reproduce it from number of patients you have at Columbia, right? So I think, and that's the way to go, is to have a process being replicated at multiple places possible so that it's reachable to as many patients.
So, one of the questions is right now, if someone is interested, they can reach out to you, and we have that information in the chat. They'll reach out to your clinical team to see if they qualify. But a challenge, of course, is someone needs to go to where you are. In Bethesda and have the work done there, which is why we need multiple places. And we're working as closely as we can with you and as quickly to establish this in New York and beyond. So Stanley, I have a question for you. What is the surgery for this going to look like? Is the patient going to be, in and out in an hour? Is it going to be a week in the hospital? What does it look like?
Oh, I think the surgery will be done as an outpatient. It'll be done under local anesthesia and will take, I estimate, between 1 and 2 hours. A normal vitrectomy would be done, small gauge instrumentation. And the only difference is that a new instrument would be developed that would be used to insert this layer of cells directly under the macula. A small cut would be made into the retina, and this sheet of cells would be slid under the macula. And it could, with experience, it could probably be done in an hour with The first few cases would probably take a little longer, an hour and a half or so, to make sure everything was done properly.
So would you both say that this really is analogous to a cataract replacement?
I think
Or do we get there?
at some stage we will get there. So remember when cataract surgeries, how they were done in the past, and Stanley can tell you more about this. You would just knock off the lens in the eye and let it just fall back into the cavity, and that's it, right? But we have come a long way from cataract surgeries as well. I think this will get there at some point, but maybe Stanley can comment more on it.
We don't know how it's going to be much more complicated than that, Susan, because we're taking these cells that are grown in your lab and then implanting them in the eye. So there's a lot of testing to make sure they're safe, number one. And secondly, in the transportation and the processing, we don't want to damage them. So we have to make sure all procedures are totally closely monitored to make sure that these cells are healthy when they're injected into the eye.
Well, I think that's an important point, is the process here is so unbelievably important. I think, one of the exciting things that we have to draw on is Pete Coffey, of course, in London, I think about 8 years ago, was able to, in what was just supposed to be a "is it safe" trial, restore vision in two elderly patients. He said to his chagrin, they were able to regain their driver's licenses. It's all of those steps to actually get it to the point where it's, it's a routine standard of care, and you have macular degeneration, you have dry AMD. This is what you do. So what do you think are the biggest challenges for us to overcome? And we're working as quickly as possible, but I think it's important for people to understand all of the things that are really kind of the lessons that you've learned along the way in both your practices and now, Kapil, in actually recruiting patients for your trial.
Yeah, so I think in terms of challenges, this is like, as Stanley said, we're still learning a lot about these cells, how we, quality control them and make sure that we are delivering the right kind of cells. But to really make it a widely available technology, I think, or a cell therapy, there are many things that we have to do. And some of these include really making the process consistent across multiple sites, right? So right now we're doing here at NEI and in your site, but if we want to do it at 20 different places within the country, how can we ensure that it's consistent, right? So things like adding automation, removing subjectivity, using artificial intelligence to QC. So all of this will not only make it more streamlined, make it more successful, also reduce the cost as we move along, right? And right now, as we transplant, we are doing live transplants. So that means the cells are brought live from the manufacturing site to the clinic, but that's not in the long run sustainable. If you want 20 sites, or if it's one place and deliver it to 20 hospitals, you want people to cryopreserve. And just like, as the lenses are delivered now in a pouch, you just open them and load them and deliver them. We are working on preloaded RP patch in a surgical tool, in a delivery tool, frozen as it is, so that when it reaches the surgeon, they have to thaw it and then check that it's there, it's intact, and then transplant. So this is the future of the technology, where we have to get it so that it's available to masses.
I think that's a really important point. I think we do want this, and we do believe that this can be a therapy for a routine therapy. But in order to do it, the cost has to be down. And as we've seen, even with the COVID vaccines, where the pharmacists had to get used to drawing out individual doses, right? As opposed to just taking a pre-filled syringe. You want to get to that kind of pre-filled tool that delivers the dose as soon as you've correlated that it's correct for the patient. So I guess a question I have for both of you is, why is the eye a good place for these early stem cell therapies? I mean, why start here?
Stanley, do you want to start?
Well, I think there's a thing called immune privilege, where in certain spaces, the immune system doesn't attack tissue that's not not natural. Now we're putting IPS engineered cells, so hopefully it's the same patient's uh, histocompatibility factors, and they'll be won't be recognized by the eye. But once it's in the eye, you can see what's going on much better than any other organ. If you transplant into the body, you can't see what's going on unless you have an X-ray, or and it doesn't have the resolution. If you look at our technology, we have the resolution to look at micron changes, so that we can see early changes, very, very early changes, and detect them before the patient even might uh, have any symptoms.
It literally is a window.
So
Yeah,
exactly, yes.
yeah,
Eye is a window and such an easy accessible organ
yeah.
that now when we're testing these transplants in animals, we can literally go every single day and look what's happening in the eye. And like Stanley said, the efficacy of the transplant, when we're looking at it, we're looking at almost single cell resolution. So that level of technology is not available yet for other organs. And then the other side of this is manufacturing. So RP cells were one of the first cell type made from pluripotent stem cells. And it was perhaps serendipitous because these cells are pigmented. They saw those pigmented cells in a dish where the stem cells were differentiated. But be as it may, this is where it started, right? So when people saw, oh yeah, we can make RP cells from stem cells, we can expand them. That's where the first protocols of making RP started. So there's many factors that have contributed to this kind of eye being the first organ, RP being the first transplant. And I think this combination of uniqueness of eye and uniqueness of RP as a tissue, easily amenable from stem cells, has helped us.
So, I think one of the questions from the audience is, how do we know that these cells are going to survive? And that's the biggest challenge for us as clinicians and scientists, because the cells became degenerated for some reasons that we don't completely understand. So when we put a new, healthy, young layer of cells back into the eye, are they going to survive? And to me, that's the biggest question, and that's the biggest challenge. Kapil, what do you think?
Well, the work we've done in animal models, we've managed to keep these cells for months easily. And these are xenocells in animals. So animals' immune system is trying to attack them and trying to get rid of them. And despite that, for months, we could keep these cells alive. So I have no doubt. And then the other aspect of this is, if you look at the early proof of concept surgical procedures that were done, where people would cut out RPE from the periphery of the same eye and translocate that into the region of damage. They saw those when the transplants were successful.
Yes.
Those survived for years, right? So
Yeah.
they have 7, 8, 10 years of data now.
Yeah.
And I think that provides the proof of concept that stem cell transplants, if delivered correctly, would have a fighting chance of surviving in that area.
And
So
maybe those healthy cells will recruit the cells that are borderline and somewhat damaged to become more normal.
yeah, that actually kind of the effects of the cells themselves, because they're not inert.
Yeah.
I think that's another really fascinating thing. So the initial trials, they're safety trials, phase 1, 2. How soon do you think we'll know if it works? I know, in Pete's case, from a patient's perspective, he expected only to make sure that he was doing no harm, but instead, somewhat miraculously, over a very short period of time, the site was restored.
Yeah, so I would say these are phase 1 to a safety feasibility trials, right? So in Pete's case was unique. The kind of patient population they picked had an acute drop in vision, and by transplanting at the right time, they could recover that acute drop in vision. The kind of patients we are trying to target here, between what Stanley's trying to and we are trying to do at NEI, are dry AMD patients where the vision decline is much slower and happens over a longer period of time. So at early on in early patients, we're only going to be looking at that the cells are safe, it's feasible to do it. We can follow these patients for a while to see the cells are alive and they're not causing any harm. I think it would be, it's too premature to call it that the cells in dry AMD patients will immediately start showing any signs of recovery.
Right. One can always hope, but we'll see. So, what are the restrictions that you have? I know we have the complete information, but in general, on age or underlying conditions for this therapy. I mean, we have to be more strict in the trials, but do you see this ultimately being able to be, offered to, someone in their in their 90s? And what about underlying conditions?
So at this stage, of course, we're being careful at the first few patients because we don't want any confounding factors messing up our outcome variables. But I think if we, as we move forward and we have demonstrated that the transplant is safe and we can routinely do it, I think we can easily offer it to a broader population of patients.
And one of the questions I think that we have is, this is just the beginning, right? I mean, there are many causes of blindness. If this trial is successful, I'm optimistic, but also this is very rigorously designed. I believe it will be successful. We have the question, of course, is it a one and done, right? Is it going to last? What's the durability? That's the same, question we're asking with our COVID vaccine immunity. How long is it going to last? We don't know. What about the other, sort of conditions, that cause blindness that are down the road? Is this a path forward into those as well?
Absolutely. So first of all, I do want to say that we are hoping that this is one and done. We are looking at this as a replacement cell therapy to replace those damaged RP cells in the back of the eye that Stanley was showing in the AMD patients. Now, whether it will restore complete vision, that will depend on where we transplant, because if the photoreceptors are already gone, RP transplant is not going to recover the photoreceptors, right? So then the future is, as the question you asked, is now to develop a dual RP photoreceptor transplant. And we're working towards it in collaboration with our partners in University of Madison, Wisconsin. So to have RP photoreceptor sandwich transplant. So that's definitely the next step, which should be done and is being done. RP transplant itself can be useful for also many other monogenic conditions, many forms of retinitis pigmentosa. We're currently testing it for choroidemia, for instance, in my group, where we hope that RP transplant at the right stage can recover both the choroid and the photoreceptors. In many monogenic conditions, as you know, the challenge, unlike AMD, which is a disease where many genes and many risk factors are involved, in monogenic conditions, it's a single gene that causes the mutation, leads to degeneration of the cells. So the problem there is that even if you made the RP cells from patients on iPS cells, they still carry the mutation. So one would have to correct that mutation first before going to make RP cells. But that technology is already there. CRISPR-Cas9 just got a Nobel Prize last year. So that technology is already going into the clinic. And what we will do is, and we're working towards, is to do an ex vivo gene correction at the iPS cell stage of any form of retinitis pigmentosa patients, correct the gene, make RP cells, and transplant. And if we need RP plus photoreceptors, well, that's what we're already working towards as well. Right?
So, diseases like, myopic macular degeneration or Stargardt's glaucoma. Do you think, Stanley, stem cells will be applicable to these conditions as well?
Well, certainly myopic macular degeneration is not too different from dry macular degeneration, so it could be helped. Glaucoma, possibly, to help regenerate the retinal ganglion cells. I mean, that's already being explored in some animal work, but so certainly that could be, those could be possibilities.
Do you think there are any risks or downsides in a cell therapy for AMD?
Well, the risk always stays until we have proven that the cells are safe, right? The risk is always there. There's a risk, of course, of the procedure itself. It's not a simple injection. It's like Stanley showed, it's a little bit more than a minimally invasive procedure. You're going under the retina, delivering the transplant. There's some risk associated with giving immunosuppression, which we took care of by using autologous transplant. But iPS cells, pluripotent stem cells themselves have this inherent risk. What if the cells are unstable? What if their genome is unstable? What if they become oncogenic? And I think we've done, as you said, we've designed this so rigorously. We did so many tests. We transplanted, so many animals to demonstrate to the FDA that these cells do not become tumorigenic or are not toxic. But that is the result as it is with animals. Unless we do the first few patient transplants, we'll never be sure that this transplant is safe. So that inherent risk is there, but I think, we do the right science and let the science guide the safety of the transplant. That's the principle we follow.
So I think, Stanley, you were talking before about basically, being pioneers.
Yeah, the risk of the procedure is maybe 1 in 2,500 of infection, which is the typical infection rate after vitrectomy. And the other would be retinal detachment, that when you make a small incision and slide something there under the retina, you could have a retinal detachment. So in general, that can be repaired. So hopefully they wouldn't lose vision.
So hopefully this is a, low to medium risk, maybe low risk, particularly because, it is the eye, it is accessible. Things can be, repaired if something goes awry. And I think it's incredibly exciting to know that we really now are on the path to this cell therapy. And something, Stanley, that you mentioned, which is, not a primary objective of this trial, but I think of either of the work that we're doing or Kapil, the collaborative work that we're doing with you, is to see what effects simply having these, these kind of newly created cells in the eye, what effect are they going to have in terms of, helping to send out these regenerative signals, if at all? I mean, that's a very interesting question.
Yeah.
It certainly is, and we are getting some glimpses at that. Like I was just saying, we are trying to test this transplant for choroidemia. Choroidemia is a condition where the choroid, the blood vessels behind the back of the eye, also start to degenerate. So we have an animal model where similar things are happening, and we are seeing that over the area of the transplant, these RP cells are able to regenerate the vessels as well. So those regenerative signals are spreading to behind the eye as well. So we definitely think that this will happen.
But now, similarly, do we think they're going to go into the wrong place, or are they going to stay where they're being put? I mean, I guess it's sort of a question of how are they going to integrate into the retina and establish the connection to transfer the signals to the brain, but not go someplace else?
Yeah, no, again, excellent point. So this is, again, goes back to part of the safety studies that we did. So the beauty of transplanting an RP patch, the way we're doing it, a single monolayer of cells, these cells are tied to each other. They're not freely walking around. So they stay in one place because of that. And we demonstrated that in many animal models, that once we transplant and follow them for months, these cells are staying in one place. They're not migrating. And then the second question you asked is, what about the connections to the brain? Well, luckily, these cells, even though they're part of the eye, they don't form synaptic connections that the neurons need to talk to each other. These cells interact with the photoreceptors just through little hair, like you can see in the back here in the picture that I have. These cells have little hair on them, right? So they talk to the photoreceptors with those little hair, and that's all they need. And we've demonstrated already, both in a dish and in animals, that their cells function by transferring their signals to the photoreceptors and to the choroid. So we were quite optimistic that cells would do the right job they're supposed to do.
For those of you who are lay people, you can see that Kapil's background, there's a layer of those RP cells blown up in high magnification. So you can see how those RPE cells cluster together and those little fingers all extending around the rods and cones of the eye.
Yeah, so each of this structure is one cell. Each hexagon that you see above me is one cell, one RPE cell.
So, Kapil, have you actually transplanted anyone yet?
Unfortunately, no. Right after we got the approval to transplant patients, pandemic started and we were shut down for months, as you know, as many other places were.
Mm.
And since these are elderly patients, we were also very cautious to not uh, put them on an undue risk of COVID by bringing them in from different places. So now that most of our patients are vaccinated and our clinical center is back up and running, we are getting ready to do the first transplant, hopefully this spring, and more to follow in the next months.
So this is an opportunity for anyone who is interested and willing to travel to the Bethesda area, or perhaps is there already. This is a wonderful potential opportunity for you. So I'd like to close by asking each of you what makes you optimistic about this therapy and its ability to restore or stabilize vision in AMD patients. I mean, we have all together been working a very long time to bring this to the point where we are now, where we're just, kind of working on the process logistics and working in our case, and in your case too, I guess, with the FDA to push this through to patients. But it's taken, you've devoted a lot of time, a lot of commitment. So what's behind that? What is your optimism? Stanley, should we begin with you?
Sure. I'd say, when you think about the history of medicine, transplantation has revolutionized what we can do for patients. And so this is just another step of the transplantation process, a microcosm of large organ transplantation, that you're transplanting a layer of cells and it actually regenerates the abnormal function. So I'm pretty excited about this technology and hopeful that it will at least give some regenerative capacity for our patients and restore quality of life.
Thank you. Kapil, what do you think?
First of all, I couldn't agree more with Stanley. It's absolutely right what he's saying. Second thing I want to say is that I think regenerative medicine is at those crossroads, that we have built that critical mass of enough proof of concept studies that we can do this, especially in the eye with RPE. And in the last couple of years, if you notice, venture capitalists and pharma have invested billions of dollars in regenerative medicine because they've realized that this is the next future. Regenerative medicine is a future of medicine. How Stanley is saying that this is going to transform all those diseases that we could not treat so far, right? And I think that's, to me, that is really optimistic. We're working on things like, complete closed system from beginning to end with automation, using artificial intelligence. These are the technologies, these disruptive technologies are what's going to revolutionize this field completely and help us bring this technology to cell therapy to every single patient.
Well, we are all drinking the same Kool-Aid, and I think it's in my view, this is clearly going to bear fruit for patients. I'm incredibly excited about it, and I want to thank you so much for joining today and providing so many insights for everyone who is with us. And please, again, for those people who are interested right now, make sure that you go to our chat now. You can reach out to Kapil, and if you're interested in supporting the work that we're doing to make this happen, please let us know. We'd be delighted. We always need more support to to get this done. So thank you so much, Stanley and Kapil, for your time, and thank you everyone for joining us. So
Thanks, Susan.
Thank you, Susan.
all
Thanks
right,
for bringing us together.
absolutely. Okay, look forward to catching up very soon.
Thank you. Bye.